Can we see that first slide?
We can indeed, the soil sociobiome.
Good.
Yep.
Okay, soil sociobiome. Right, we're away.
Excellent.
Okay, so this is part one. I'll also be speaking on Friday about the soil sociobiome, and the sociobiome is basically the interaction of all the different microbiomes in the soil. And today I'm going to be talking about what is in a seed and why does that matter. So if we look at seeds, most of us who are familiar with seeds can instantly recognise that they are representative of different plants. In other words, like a bean is always going to have a bean seed and a tomato is always going to have a tomato seed. But what is actually in those seeds and why does that matter? So what's in a seed? Well, just in very general terms, it contains the genetics of the plant. It contains some energy and nutrients usually. And it also contains microbial genes, which is the thing that we've probably overlooked in the past. So plant genetics, we're all very familiar with the fact that the plant DNA determines what kind of a species it's going to be and what cultivar or variety it's going to be. We're also aware that most seeds, not all, but most seeds contain some kind of starch or oils or proteins to provide energy and nutrients for the new plant. What we haven't really been aware of until relatively recent times is that within that seed, there are also microbes, which is the core microbiome of the seed, and that these microbes that are encapsulated within the seed have a large influence on the successful germination and the establishment and the growth of that plant. So in the past, we've tended to consider only the genetics of the plant. You know, we wonder about when we're talking about different varieties of something that we're going to plant, like will it be more drought tolerant or more frost tolerant or have a longer growing season or a shorter growing season or all those kinds of things. We look at that as being the genetics of the plant, but in actual fact, the plant DNA, which will determine all of those things, and the microbial DNA together constitute the plant hologenome, which functions as a unit. And in future plant selection and plant breeding, we actually need to take notice of the microbial DNA as well as the plant DNA because it is extremely important. So the microbial DNA is actually stored in what's called the plant's microbiome, and the core microbiome of every plant will have keystone microbial taxa. In other words, what's in a tomato is going to be different to what's in a corn plant. It's going to be different to what's in a wheat plant or a ryegrass plant. Each of those broad genomes will have keystone microbial taxa that we can use to actually identify them. And these are going to be important for plant fitness. But the most important thing about this is it's not just an academic, it's not just purely esoteric, because we can enrich that core microbiome and improve plant fitness, or we can deplete it over time, depending on how we actually manage our plants and how we manage our soils. So this is going to be very important if we want to optimize plant production into the future, and also if we want to use less inputs, is to actually understand the importance of microbial, the microbial contribution to the fitness, the function, the productivity, and the tolerance to stresses, the tolerances to pests and diseases, and all of those things are very much regulated by the core microbiome of the plant. So the core microbiome consists of bacteria, archaea, fungi, and protists. They're quite diverse, and there's quite a lot of species involved. This is just an overview of the life cycle of a plant, I suppose, from, well, if we start with A down here of a germinating seed, and as it grows, matures into a plant, it'll bear its leaves, etc. And all the little dots that you can see inside those leaves and around the roots and everything represent the microbes. So there's going to be microbes on the leaves and in the leaves and on the stems and in the stems and all around the roots. We're very aware of the rhizosphere microbiome. And then eventually that plant will produce seeds, and then the microbes that were the core microbiome that was in the plant will be encapsulated in the seeds, and then the whole cycle starts again. The interesting part of this diagram is this section here, because this is where we can have a gain of function, if you like, of extra microbes being taken into that plant at this point, depending on how we manage it, or less microbes being taken into the plant depending on how we manage it, and therefore carrying through into the microbiome of the next generation. So if what happens here is something that has a degrading effect or a toxic effect even on plant microbes, for example, using a fungicide is going to knock out key fungi that need to be associated with that plant, but also insecticides and high analysis fertilizers. All of those things are going to affect the microbial, the microbiome of that plant, and therefore are going to affect what ends up being the core microbiome of the seed, and the subsequent generation of plants is going to be at a disadvantage because it's now functioning with less than optimum core microbiome. So the microbes that we're interested in when we're talking about plant microbes, they live everywhere in the plant. They're in the roots, they're in the stems, the leaves, the flowers, the fruits, the seeds. All plants have microbes in all parts of the plant. And what do those microbes actually do in there? Well, if we just look at this, this is just a magnification of that section of the diagram with the seeds. You can see the little different kinds of microbes there in the seed. When that seed begins to germinate, those microbes actually move out of the seed and into the soil. It's sort of like, I've used this diagram because they explode out into the soil. And really they're just like scouts. They're going out there on a reconnaissance mission, and they're going to bring information back to the plant. And is it like, well, have we landed on rocky ground or are we in fertile soil? And who else is here? Microbes very quickly need to establish what other microbes are around them, what kind of soil conditions that they've found themselves in, and other things about the environment as well, like is it moist or dry, is it hot, is it cold? All these sorts of things are actually going to be determined by the microbes that move out of the seed, but in particular they're going to interact with the microbes that are already resident in the soil. And then both the microbes that were in the seed and moved out of the seed and some of the microbes that they come into contact with in the soil are going to move back into the growing plant and become endophytes in that plant. Now, at this point, this is a very critical point in the life of a plant, because at this point, if we have things like fungicides around that seed, then obviously we're going to dramatically reduce the diversity and the abundance of microbes that are able to move back into that plant and assist it in its growth. So as the new plant grows, the microbes that it started with and the new microbes that it acquires will be very important for its fitness. So when we look at a germinating bean seed, all we see are basically, you know, what is visible to the human eye. We see a seed, we see some roots, we might see soil sticking to the roots, which is a really great sign, and then we'll see the emerging cotyledons. But we have no idea of the complexity of the microbial interactions that are going on around that plant because we can't see them with the naked eye. The microbes that were in the plant that move out and then move back in, and also some of the new ones that move in from the soil, are called endophytes. And an endophyte is just, endo is inside and phyte is plant. So any microbe that lives inside a plant is called an endophyte. Endophytes are beneficial and they colonize the internal plant tissues of for all or part of their lifetime. In other words, they move in and out of plants all of the time. So when the seed germinates, the core microbiome will move out into the soil, form relationships with soil microbes, they all move back in, and then over the rest of the life of that plant, there will be microbes moving in and out of the plant depending on what the plant needs, mostly through the plant roots. So what are these endophytes important for? Like, why do we need to know about endophytes? They're very important for nitrogen fixation. We now know, we're fairly familiar with what happens in leguminous plants and that there are endophytes inside the nodules on legumes, but we now know that they're very important for nitrogen fixation in non-leguminous plants. So we are seeing a lot of farmers in Australia now planting multi-species forages, for example, that don't have any legumes in them. And when you do a tissue test, you find that things like chicory and plantain have excellent, like luxury levels of nitrogen in their leaves, but there's no legumes in the system, there's no nitrogen being applied. So where is it coming from? Well, it's coming from the atmosphere, because the atmosphere is 78% nitrogen, but something has to fix that. So the microbes that are fixing that nitrogen are actually called endogenous or endophytic diazotrophs. That would be a conversation stopper at a dinner party. But endophytic diazotrophs, so endophytic, they live in the plant. So we're not talking about free living microbes around the roots. We're talking about microbes that are in the plant itself, and a diazotroph is something that is able to fix atmospheric nitrogen. So the advantage of these being in the plant is that they don't have to compete with other microbes in the rhizosphere. They get plenty of energy from the plant to break that triple bond in the nitrogen. And also they can find locations within the plant where there's a low partial pressure of oxygen, which they need for the reductase enzyme that's used for nitrogen fixing. So that's just one thing that happens when we actually have sufficient endophytes in our plants. As I said, there's a lot of things that we do in modern agriculture that reduce the amount of endophytes or the number of endophytes we have in plants. But if they're there, then they can fix nitrogen. They can also produce phytohormones, and these include, all of the things that many of you will be familiar with, the auxins, the cytokinins, the gibberellic acids, etc. They're important for nutrient acquisition, because sometimes plants will take things up, like even phosphorus, for example, may be taken up in a form that's not available to plants, even though the plant has taken it up, and we'll find that it can be rendered plant available actually inside the plant by some of these endophytes. Gives plants incredible tolerance to abiotic and biotic stresses. So abiotic is something that's not biological, so it's going to be like a temperature stress or a moisture stress or something like that from the environment. And a biotic stress will obviously be a pest or a disease, and it provides defences against pests and diseases. So the metabolic pathways and the things that happen actually at a biochemical level in plants to confer all of these advantages. So I'm not going to speak about those, but there is a lot of literature on this, and if anybody wants to read about it, you'll find it's quite extraordinary actually what microbes do for plants. And the interaction between the host plant and the microbes that colonizes is a continuum. It doesn't have a set beginning and it doesn't have a set end. I mean, what comes first, the plant or the seed? And the plant and the seed are going to have microbes in them. And the other thing is that because it's a continuum, it is something that plants will react to stresses by recruiting more microbes from the soil if they need them. So let's take drought as a typical example for us in Australia anyway. If a plant is growing in a situation where it has sufficient moisture, it will have a certain endophytic microbiome. In other words, there'll be certain microbes living within that plant. And then in a drought situation, if you check inside the plant, you'll find it has a completely different set of microbes in it, because it has recruited microbes from the soil that it needs to help it to deal with drought. And then when moisture becomes non-limiting again, it changes its microbiome and no longer needs those ones. So this is a continuum that just goes on and on and on for the life of the plant. There's probably too much detail in here for me to be able to explain it, and I'll just go over it very, very quickly. But we have the seeds here that do have the microbes within them. They germinate. At that stage, some of those microbes will actually exit out into the soil and perhaps stay in the soil and not come back. And then there'll be others from the soil that will come in. And we go through to the seedling stage, and then right through to the flowering stage, and then the reproductive stage of the plant, and then back to seeds again. So all of this is a continuous cycle, and at any point in this cycle, we can have microbes entering and exiting plants. And most of these movements of microbes and most of the function of microbes is actually determined by quorum sensing. This is critical to the whole movement and the whole mechanism of microbial activity. So just back to the original, the diagram that I showed you before, it again is the same thing. We don't really know where this starts and where this ends. It's a cycle that's gone on for millennia, basically. And the point being that over time, plants can actually suffer a degradation of their soil microbiome, or they can have their core microbiome enriched, depending on how we manage them. Plants contain more microbial cells than plant cells, which is not unusual because the same situation happens with all living things. So humans actually contain more microbial cells than human cells. It's quite normal for that situation in a holobiont. In other words, all of us are dependent, all living things are dependent on microbes. But microbes are not able to see, hear or speak, and they use biochemical signals to communicate with each other and with the seed. So at that point where the microbes leave the seed, move out into the soil to explore the environment that they're in so that they can react to that environment, depending on whether it's a very low fertility environment or a high fertility environment, will depend on what the seed does next in terms of recruiting microbes. And all of that communication that goes on between the microbes, between themselves and also between them and the plant, and between the plant and the microbes, is all done using biochemical signals called autoinducers. And when the concentration of these autoinducers in the environment reaches a critical level, they can regulate gene expression. So the extraordinary thing about microbial signalling is it can switch genes on in plants and it can switch genes off in plants. So if we don't have microbes reaching a critical level, a lot of very important genes in plants will remain switched off. And we need to know about that because those genes are important for plant productivity, for plant tolerance to stress, and for plant resilience in the face of things like climatic extreme, but also to pests and diseases. So this is termed quorum sensing. I have talked about quorum sensing before, but basically it's density-dependent coordinated behaviour. In other words, it doesn't happen unless you have sufficient microbes there for it to happen. And it occurs in all species of bacteria, archaea, fungi, and viruses. All microbes use quorum sensing to regulate their activities. That's just an example of some of the quorum sensing molecules, the autoinducers. They're basically chemical rings with different side chains, and the different side chains send different messages. And they're going to be generated by, say, a bacteria here. They go out into the environment, and then they need a receptor site in order for the message to be accepted and translated into action. And then once we have sufficient of these in the environment, then various genetics genes will be targeted and activities can take place. So endophytes, these are the microbes that live inside plants. They use quorum sensing to coordinate their activities, to regulate plant growth, stress tolerance, and resistance to pests and diseases. So basically, it's quantum biology for soils. But it's not about adding microbes to plants or to soil. Lots of people have, when they've realised how important microbes are, they've thought, oh, let's add some microbes. And of course, there's been lots of companies that have wanted to produce microbes, sell them to farmers, etc. As a general rule, there's a very, very low survivability of any microbes that are added to plants or to soils because they have to compete with the microbes that are already there, and they're generally not very successful in that. So it's not about adding microbes to plants or to soil, but to actually enabling the ones that are already there. All of our seeds have a core microbiome. The soil has most of the microbes that are in soil are dormant, and they're just waiting for plant root exudates to basically wake them up. So everything we need is there. It's all about enabling this relationship between plants and microbes that has been there for millennia to actually enable that to function more effectively. So when microbes move from the soil into plants, or particularly through the plant roots, that's termed biological induction. And basically what we see here, corn plant here with the roots, and just showing the different coloured, different kinds of microbes that can move into the plant through the roots. We can also have microbes move into plants in through the leaves, in through the flowers, but as a general rule, most of it occurs in through the roots. So what are the factors that are going to improve that process of biological induction, and what are the factors that are going to be inhibitory to biological induction? Well, we know that it can be increased through plant diversity. If we have a minimum of 4 plant families, especially, we see that rates of biological induction markedly increase. Can also be increased through the use of a biostimulant, which is not adding microbes to plants. It's actually just stimulating the microbes that are already there. So plant diversity significantly stimulates biological induction. I did talk about this to some extent last year, where we have, in this case on the left-hand side, we have a plant that's in the bean family, which is Fabaceae, and on the right-hand side we have a rice plant, which is in the Poaceae family or the grass family, and they have very different microbiomes. This one's shown as a green microbiome around the rhizosphere, very different rhizosphere microbiomes, I should say. This one has a blue rhizosphere microbiome. They also have microbes all through the plants. So they have a philosphere microbiome, which are the microbes that live on the leaves. But the endophytic microbiome is the one inside the plant, is the one that we're interested in. Now, if we have a paddock that's just all full of beans, for example, all of their rhizosphere microbiomes are going to be the same. And if this microbiome determines that the one next to it is the same as itself, then they will compete. So when all of the plants that are growing together are from the same family, they will compete for resources because they sense that they are very similar and they're using the same resources at the same time, at the same root depth, the same kinds of roots, everything. That similarity actually results in competition. But when the microbiome ascertains that the microbiome of the plant next to it, again, this is all going to be done through microbial signalling, through chemical signalling, then they will cooperate. So the more difference that we can have between the plants that we put together, the more different those plant families are, the more different the microbiomes are, the more likely they are to compete, to cooperate, and that's when we get what we call over-yielding. But the significant thing that I wanted to mention today was that when those plants are growing close enough together for their roots to actually mingle, or for them to be joined by a common mycorrhizal network, which they will be if we're not using fungicides, then they can swap microbes, they can swap genetic material. So if this plant, for example, needs something that is not in the microbial population in its own rhizosphere, it can borrow it from its neighbour if they are different enough. And that's why we're starting to see really dramatic things that are very difficult to explain when we put plants from different plant families together in communities. It's because of this biological induction that is enhanced by dissimilarity of plant families. So when we put very distantly related species together, there is a much larger microbial gene pool. Remember that when that plants take up a microbe, they're actually taking up the DNA of that microbe. So they're taking extra genetics into their own, so that their hologenome is their own DNA plus the DNA of any microbes that they've got living within them. And they can use that microbial genetic material for things like nutrient acquisition and immunity to pests and diseases. And we're seeing many, many examples of that taking place these days, which is great. So 85 to 90% of plant nutrient acquisition is microbially mediated. We already know that. So nearly huge amounts of fertiliser, synthetic fertiliser like high analysis fertiliser, things like urea or MAP or DAP, huge amounts of that are wasted because it actually has to go through a microbial intermediary to be taken up by the plant anyway. And the efficiency rates of synthetic fertilisers are very, very low. And what is not taken up by the plant ends up in the environment as a pollutant and is very detrimental to the environment, as well as costing farmers a lot of money because it is not an efficient way for plants to acquire nutrients. So this experiment, this is from a cover crop demonstration trial in Ontario in Canada. This is just a monoculture of ryegrass. Monoculture of radish, which has actually received a base fertiliser. It has been fertilised, but it's looking very nitrogen deficient. And just to the right of that, these trial areas were about 20 metres across. So just to the right of that is the same cultivar of radish, but grown with plants from 3 other families. So we have 4 plant families here. We have the radish, hard to recognise because it's a totally different colour to the previous photo. We have oats, which are in Poaceae. So radish is in Brassicaceae. We have oats in Poaceae. There's a bit of Phacelia here in Boraginaceae and a sunflower in Asteraceae. So we have 4 plant families growing together, and all of a sudden there is no nutrient deficiency. In fact, if you measured the leaves of these things, which is the sort of work that we've been doing in Australia recently, there aren't any legumes in that mix. But if you do a leaf test or a tissue test on the leaves of these plants, like the sunflowers and the Phacelia, etc., you'll find they have luxury levels of everything, and you can tell by looking at it that they have luxury levels of everything. So that's just the difference between growing something as a monoculture and growing it as a polyculture, where we have very different plant families. This is a microbial effect. This is just straight biological induction and microbial. Oops, nothing there. So this was a little photograph that was sent to me by Chase Thornhill. He's in California. It's a wine grower, and he had bought some seeds from Green Cover Seeds in Nebraska to plant in the interrows of his vineyard. And he got 12 different kinds of seeds, but he didn't really know very much about cover crops, and he didn't know what those plants looked like. So he bought some little pots from the from the hardware store, and he bought some potting mix, and he planted each of the seeds in the little pot just so he could identify it when it was in his multi-species interrow mix. Now you can see that when those seeds were planted without any fertiliser into potting mix, they really didn't grow very well. So here's our 12 different kinds of plants, even things like this one at the back here is faba bean, which you would think, because it should be able to fix its own nitrogen or associate with rhizobia to fix its own nitrogen, shouldn't really need to be fertilised. And none of them really grew very well at all in potting mix. And then he had a larger pot that he put all 12 species together in this larger pot, again with no fertiliser, and they're really crowded in there, and they're all growing perfectly well. And I thought that's just such a classic example. I mean, it's not randomised, replicated, top level science, but it's just, it is just so obvious to me that these plants need to be together, and that when you put them together, that all kinds of incredible things happen that don't happen when you grow them on their own. So why is the plant's core microbiome, that is the microbiome that's encapsulated in the seed, important? I mean, the subject of today's presentation is what's in a seed and why does it matter? Well, it's because the seeds carry not only the DNA of the next generation of plants, but also the microbes that will live in association with that plant. And this intergenerational transfer of endophytes by the seeds is called vertical transmission. So again, we just come back to this diagram here of how things are carried through from one generation to the other, but what we do at the plant stage, when there is actually a plant there in the field, how we manage that plant is going to determine what happens or what ends up in the core microbiome of that seed. So the way we manage the soil environment will determine whether the plant's core microbiome is enriched with each generation of plants or whether it's depleted over time. So often I hear growers say that they've imported some seed, maybe it's, certified seed of a certain variety, and they may even be in the business of producing certified seed themselves. But they'll say that in the first year, when they grow that seed, because it's come from an environment where it was grown with a lot of high analysis fertiliser and was grown as a monoculture, et cetera, that it really doesn't grow very well on their farm compared to other seeds that they have growing there. And then if they're using things like compost extracts or vermiliquid or whatever, in other words, they're growing those plants in a biological system, they're not using high analysis fertilisers, each generation where they keep the seed and replant it, they will see that each generation is better than the one before. The plants germinate faster, they're much less prone to any kinds of pests or diseases, and they're more productive, they produce higher yields. So that is because the microbiome is being enriched with each successive generation. And the indications are that when we use high analysis fertilizers, it actually depletes the plant's core microbiome over successive generations, because when that seed germinates and finds that it's in an environment where there's, well, probably too much available nitrogen and too much available phosphorus, there is no need for it to form any kind of relationship with soil microbes in order to obtain those things, and so it doesn't, and so that you end up with a very poor endophytic microbiome in that plant. And then that passes through to the seed, like depleted core microbiome passes through to the seed and then through to successive generations. So if we have management practices that enhance microbial abundance and diversity, this will actually enrich the plant's core microbiome over successive generations. So I'll leave it there because we do have another speaker after me, and also I wanted to leave a few minutes for questions. There was a few other things I was going to talk about, but I will carry them over till Friday and talk about them in my presentation on Friday. So I'm fine for questions, Kevin, if you, or John, I'm not sure who's facilitating questions. John, I guess.
Yeah, that's right, Christine. Listen, thank you very much. That was terrific and enlightening, and I certainly have a couple of things I'd like to ask you myself, but let me instead go to our questions. We've loads of questions in. The first one is from Janine Hazelhurst, and she asks, she says, for maximum carbon sequestration, you recommend 16 plus species, 60 plus for dairy. Do the 16 plus species exchange microbiota and increase beneficial growth?
Who recommends 16? I recommend 4 plant families.
Okay, this is...
You have to have a minimum of 4 plant families. Like you could have 16 species of grasses or 16 species of legumes, and you've just got 1 plant family.
Okay.
So if you have 4 plant families, like I showed an example with a sunflower, a phacelia, an oak plant, and a radish plant, 4 plant families.
Okay, we'll move on. Rachel Hardiman asks, she says, regarding diverse species, do they have to be sown randomly mixed in as grassland, or does it also work in mixed strips or blocks to suit arable, say, or horticultural farming?
Yes, very interesting. I mean, there's people developing techniques like relay cropping where things are grown in rows, so that you can just harvest one row or something, and then the row that's right next to it will be a different plant family, and often, well, for relay cropping, it will actually grow at a different time of the year as well, so you'll be harvesting at a different time of the year. What's happening in the last 12 months or so, or probably even 2 or 3 years, what we're seeing is a trend to what's called multi-species cash cropping, which is in a tillage situation, you would grow a whole lot of different things together, plant them at the same time, harvest them at the same time, and really the technical innovations that are needed in that area is really good sorting equipment or grading equipment, so that you need to like have an optical sorter or something to be able to separate those seeds after you've harvested them. So that, just to give an Australian example of that, one of the most commonly grown crops in this country is wheat. So if farmers grow wheat with a range of things like, say, lupins and millet and the radish, like tillage radish or something. So if they've got, make sure they've got their 4 plant families there, and try and get as much difference in seed size as they can. They'll probably put some peas in there as well because they've got big seeds, and then harvest them all together and separate them. But what they're finding is that their wheat yield is not reduced by having all those other plants in there because of over yielding, so that the biomass of the multi-species mix is higher than the biomass of a monoculture anyway, and that when you take the other seeds out, you still have a very high wheat yield. But then the other plants that you've grown in there, for example, you might put something like daikon radish in there, and there's a lot of people looking for daikon radish because they want to use it in cover crops or they want to use it in multi-species crops. So the market for that is going to be, the wheat is going to be sold onto a global commodity market, which you don't really have much control over, but it's more of a niche market for things like daikon radish and the lupins and the other things that are going to be grown in that mix. And oftentimes farmers are finding that they get more for the other species that they've put into the mix than they are for their main cash crop, but they're still growing a cash crop and still getting good yield. So I don't think that you have to make it complicated by separating out the rows, provided we have the equipment for separating the seeds once they've been harvested. And I think that's going to be one of the big technological breakthroughs that's going to happen that will enable us to have very diverse crops, is simply being able to harvest and separate the seeds.
Okay. Obviously, diversity has been one of the themes that you've pushed very strongly here, and you also mentioned along the way that the impact, say, of the use of pesticides, fungicides, in terms of presumably of simplifying, if you like, the soil structure and the biome. So can you tell us a little more about that, and to what extent, say, in the Australian experience, is that an issue, and how does it impact on I suppose, the vitality of soil?
Well, we know that fungi are very important for, as well as the functions that fungi have on their own, that we have to have a combination of, well, largely bacterial and fungal. I mean, we know that there are other things in there as well, but largely a combination of bacterial and fungal relationships in soil for it to function effectively. In other words, it can't function with just bacteria and it can't function with just fungi. You need both of them. And the bacteria that operate around plant roots, for example, they have direct effects, but they also have indirect effects. That, for example, they act as vectors for bacteria. Bacteria are able to move through the soil by using fungal hyphae as highways. I've seen some incredible videos of bacteria just streaming along fungal hyphae to move from one place to another. It's pretty hard for a bacteria to get anywhere on its own. And so if we, and fungi, as I'll mention on Friday, are actually the most important component of soil when it comes to building soil carbon. So if we knock out fungi with a fungicide, then we are having a huge impact on bacterial colonies as well, because they work in teams. Bacteria and fungi work in teams. And of all of the, like what the research is showing, and this is not just Australian research, this is mostly American research, is that of all of the chemicals that are used in agriculture, in other words, if you look at insecticides, fungicides, high analysis fertilizers, you know, the whole gamut of agricultural chemicals, the ones that have the most detrimental effect on soil, especially even on soil structure, amazingly, are fungicides. But then when you think of how important fungi are for soil structure, I guess that's not so amazing after all. So, and why are we using fungicides? You then take that one step back. Why are we using fungicides? We're using fungicides because the plant's not able to defend itself adequately. Why is it not able to defend itself adequately? It's because its core microbiome isn't doing what the core microbiome should be able to do. So the immunity of the plant and its defence against fungi is going to be largely related to the diversity and the abundance of microbes that are inside that plant. Because again, remember I mentioned that those microbes are going to use quorum sensing, and if there's not enough of them, they don't reach a quorum, they're not going to switch on the plant's genes that it needs to defend itself against fungi.
Okay.
So, you know, we're taking everything to a lower and lower level. The more we treat symptoms with poisons, we are knocking out the very things that are able to give our plants the strength to be able to defend themselves.
And of course, in the process, we're locking ourselves into, if you like, a chemically mediated system, because the very defences that plants should be able to call on have been taken out by those chemicals, which leave us then dependent, if you like, on using more of them, which I presume from the point of view of the manufacturers is quite a happy coincidence.
Yes, and it could be the reason why we're seeing this divergence between the gross value of agricultural production continues to go up, because the gross value of agricultural production includes all the input providers. So the people that are making the fertilisers and the fungicides and all of those things are making plenty of money, thank you very much. But when we look at the net income of producers, it's been over time, like in the last 80 years, it's been slowly going down, while gross agricultural revenue has been going up. So we've got this ever-widening divide between the gross value of agricultural production and net farm income. Net farm income is so low because farmers spend so much on inputs, and it may start with something that appears to be innocent, like using a high analysis fertiliser, which then alters the core microbiome of the plants, because they don't need to take microbes up from the soil to enrich their core microbiome, and then all of a sudden they have no defences against fungi and then or insects, and so we have to use insecticides, we have to use fungicides. So there's no blame being put on farmers for using those things because they use them out of necessity. You're not going to apply something and spend money on applying something unless you need it. But they need it because the core microbiome of the plant has become dysfunctional.
Okay. Can I offer to bring in either of my colleagues, Sean or John, at this point?
Yeah, thanks, Christine. I'm finding every year I meet you there's more microbes in the world. But I just wanted to ask you, is in terms of actual seed dressings or seed handling, will that have a big impact on those soil microbes, or the seed microbes?
Yeah, it will, Sean, because when the seed is germinating and the microbes that are in there move out into the soil, their primary aim really is to make contact with soil microbes and to through those quorum sensing molecules and the communication that they have, then whatever the plant needs, everything will move back into the plant and become part of like the endophytes for the newly growing plant. Well, if we put poisons around the seed like insecticides and fungicides, they are going to affect with that communication, because the communication is all biochemical signaling, and we put very intense poisons on a seed. Yes, it does affect the signaling and the communication that needs to go on between plants and microbes in the soil.
If I can jump in for a second, Christine, we have a live audience with us here in Carrigan Shannon, and if anybody would like to address a question directly to Christine, could you please put up your hand? We have a roving microphone we can send down to you. There's a gentleman at the back. Here we go. Just give us 2 ticks. Here we go.
Hi, Christine. We know the benefits of multi-species on soil biology and the different exudates and their beneficial effect on biology. Is it also possible that if you had monocultures, say, for example, of barley or wheat, but you used huge amounts of different varieties, that you could also get a biological response? Do different varieties have different exudate properties? And if we, say, for example, took grasses, and we took rye grasses, and we went with 6 or 7 or 8 species of rye grasses versus 2, is it possible that we would have a better biological activity in soil with the 6 versus the 2? And the same goes for cereals.
Could you hear that, Christine, okay?
Yes, I did hear that. That's a very interesting question. We did have quite a few farmers experimenting with that in Australia, particularly for things like with wheat, because we have probably a more challenging environment maybe here, that we could have a season that cuts out very early, or we could have a very late frost, cuts out, I mean, in terms of moisture, that suddenly it just stops raining and doesn't rain for months or something, or a very early frost, and that farmers found that if they put like half a dozen wheat varieties together, that it reduced their risks, because there was a chance that at least something was going to get through. But I think they also did notice that they had higher yields from putting different varieties of wheat together. I don't know whether there's been any research done on that. Intuitively, you would think that because there is some genetic variation between those plants, even though they're in the same plant family, that you should get some benefit from that. But you would see a much larger benefit from putting something in there that was a different plant family. And it may be something that you didn't particularly want to harvest. You might just put it in like something short, like lentils or something like that, in with your wheat. I think you might find that you get a huge benefit from actually having different plant families in there. Or the other thing is what's called the legacy effect, which is wherever a plant has been growing, the microbes that are in the soil where it was growing will remain for a certain amount of time and can be activated by another plant being put in that same spot. So that if you had like a multi-species cover crop, before you planted your monoculture. I'm talking about annual crops here rather than a pasture situation. But if you had a multi-species or multi-family cover crop before you planted your wheat, there's going to be a legacy effect of those microbes in the soil that will remain sufficiently for a sufficiently long period for your germinating wheat to be able to access those microbes, assuming that it's the length of time is not too long. So I would say, if provided it was within several weeks, that legacy effect would still hold. And that's another thing that we're finding, particularly where people have soil that really needs a lot of help, that if they plant 3 successive multi-family annual cover crops, so in other words, a cool season multi-family annual cover crop and then a cool season, a warm season multi-family annual cover crop, because all the plants are going to be switched around, they're going to be in different positions and different times, different space and time, and that you've got the legacy effect as well as the biodiversity effect and all of these things. It really, you can get quite outstanding results. But you can use the legacy effect to help you if you still just committed to growing, say, monoculture wheat, have a diverse multi-family cover crop just prior to putting that monoculture in.
Okay. On a related theme, we have a question in from Emily Grant, and she asks, how long will plant species specific microbes last in the soil, and would monocropped wheat for 20 years have enough microbes suitable for, say, a brassica crop?
I don't really quite get that question, I'm sorry. You need to have 4 plant families to have a really, I'm not really quite sure.
I imagine what she means is, if would the microbial, if you like, activity be so run down after that length of time in a monoculture, would it be able to kick in again? I presume that's the gist of the question.
Well, as I was saying, if you have really degraded soils, the best way to get them functioning effectively in a short amount of time is to have at least 4 plant families in a mix that they're all grown, like, rather than just growing brassicas on their own. Rather than going from a monoculture of cereals to a monoculture of brassicas is, yes, it's a difference. And we know that, I mean, the research shows that what's called, crop rotation, like going from different plant, rotating through different plant families, like the cereals and the brassicas and the legumes, that kind of rotation is far more beneficial for soil than just having a monoculture of the same plant family for 20 years in succession. We know that that's not good for soil. But I would really like to see people try to get away from monocultures altogether.
Of course. Now we're almost out of time, Christine, and I'm just going to wrap it up to ask my colleagues, Sean and John, if anyone has a quick final question for Christine before we wrap it up for a commercial break.
Okay, Christine, great to hear you again, and looking forward to the next instalment as well. I suppose just with relation to endophytes moving in and out of the plant, a lot of our farmers here now are crop-producing farmers. They're growing cover crops during cool season cover crops. And the question that I have for you is, one of the inputs that we use is seed, and we bring in seed from outside. So we bring seed that has been developed maybe in an environment that has a different soil microbiome to what's in our field. And I'm just wondering, from the point of view of organic and even conventional farmers, does what you're saying, does it present an argument for farm-safe seed?
I see what you mean, in terms of if you were saving seed on the farm and then replanting that seed, that over time you can enrich the core microbiome of the seeds that you're planting.
Yes.
And that's what farmers are seeing that.
Yeah.
If you were purchasing seed from somebody that was also doing the same thing and was farming it, in a way that was enriching their soils, like in a very biology-friendly way, and they were improving the core microbiome of those seeds, so that when you purchase them, that you would be buying good quality seed, like with a good core microbiome. I think in the future, people buying seed will want to know how those plants were produced, what the soil environment was, were they produced in biologically active soil, in a biology-friendly way, or were they produced with lots of fungicides and insecticides and high analysis fertilizers, which means that you're actually buying seed that has an effective core microbiome. And farmers in Australia now, I know for sure that people planting multi-species covers, for example, or multi-species forages are insisting on bare seed. In other words, seed that doesn't have any seed dressings on it, like any fungicide or insecticide. They will not buy seed unless it's bare. And the seed companies that are selling seed they just can't sell seed that's been dressed. So, and some seed companies now are actually selling seed that has has auto-inducers on it, like it has like a compost extract or a vermi-liquid or something like that already on the seed when you buy it. So it has been dressed, but it's been dressed with a biostimulant. And I think, so we've got that far. We've got to the point of people saying we want bare seed, we don't want it with the dressings on it. And now we're getting to the point of, well, actually, how was that seed grown? Wanting to know the history of it. In the same way that if you were in the animal production sphere, if you were buying a bull for the dairy farm or a ram for your sheep flock or something like that, like was this raised in a little pen somewhere where it never got out and had to walk around and graze for itself or fend for itself, or is this actually what, like we would call it, a paddock bull or a paddock ram or something that's going to be able to work out in the paddock and do what's required of it? You know, people want to know about how that animal was raised in the future, and I hope very soon farmers are going to be asked, where did that seed come from? How was that seed grown? Because they would want to know about the core microbiome of that seed, because it's going to be just as important for its productivity as its genetics. And I think in future plant breeding, we are going to have to take account of the core microbiome as well as the DNA of the seed's DNA as well as the microbial DNA. So we're going to look at the hologenome of that plant, not just its plant genetics.
Fantastic. Terrific, Christine. Thanks again, and thank you very much for joining us, and delighted that you were able to join again for your third year, and hopefully you'll be back again next year. And we're now going to take a short commercial break, and we'll return very soon with Robbie Byrne. Thanks again, Christine.